In this lesson, we'll be focusing on microbial nutrition and growth. With microorganisms, they do have to intake nutrients in order to be able to produce energy to grow and divide. So just like us, microorganisms have specific nutrients that they are not able to make independently on their own, so they have to uptake them from the environment or through obtaining them from other living organisms. These nutrients are considered essential nutrients. So essential nutrients are any substance that must be provided to an organism. So the 6 essential nutrients that microorganisms need are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, which is quite easy to remember if you just remember the acronym CHOMPS. With microorganisms and nutrients, they often require different quantities of nutrients. So if a microorganism requires a large quantity of nutrients, which is typically common if the nutrients play a very important role in the cell, navigating processes that are utilized all the time within the cell, well, then those nutrients are considered macronutrients. So nutrients required in large quantities, such as carbon, hydrogen, and oxygen, are considered macronutrients. Micronutrients are required in much smaller quantities. So if a cell only needs a specific nutrient to run one enzymatic reaction that's not continuously used all the time within the cell, it would need a small concentration of that specific nutrient, such as something like zinc or nickel. So micronutrients are required in much smaller amounts within the cell. We tend to classify microorganisms and organisms in general based on how they obtain essential nutrients such as carbon. So often microorganisms are considered heterotrophs. Heterotrophs we sometimes refer to as other feeders. Heterotrophic organisms are organisms that feed off of other living things in order to obtain essential nutrients such as carbon. So often things like fungi and bacteria will feed off of other fungi, animals, plants in order to obtain carbon as a nutrient source to sustain life. I mean, technically, humans are considered heterotrophs. We feed off of other living things, such as plants and animals, to obtain our essential nutrients as well. There are autotrophs. Autotrophs we often refer to as being self-feeders. Autotrophs are organisms that can produce their own food from substances in their surroundings. So for example, things like plants and algae, they can actually get carbon from carbon dioxide in the air. They don't have to necessarily eat another organism, another living organism, in order to obtain that carbon, such as what would happen in the case of a heterotroph. So autotrophs are very much self-sufficient in that sense. And this is common for things like plants, algae, and then some bacteria, although most bacteria are considered heterotrophic. So since microorganisms require essential nutrients that have to be taken in from the environment into the cell, we obviously have to talk about transport mechanisms. So how do things enter the cell from the external environment? This is typically regulated through the cell membrane. Remember, the cell membrane is a selectively permeable layer which controls what goes into and out of the cell. So the transport mechanisms that we talk about are all dependent on a functional cell membrane. So one of the most common transport mechanisms is diffusion. Diffusion is a generic term that we use for the movement of all different types of molecules from an area of higher concentration to areas of lower concentration until an equilibrium is established. There are different types of diffusion, as we'll see here in a second. Diffusion, as we talk about it here, kind of encompasses all those different types. So essentially, you're moving things down a concentration gradient from areas of high concentration to lower concentration, which is what we're kind of seeing here in this glass. So up here we have a higher concentration of this blue dye, and then throughout the rest of the water glass, there's obviously a lower concentration of the blue dye since it's clear. So based on the rule of diffusion, the blue dye in this case, the blue dye is going to move from an area of higher concentration up here to areas of lower concentration, which is the rest of the glass, which we're seeing, until obviously an equilibrium is established. Since you're moving with the concentration gradient, since you're moving things from high to low, it's like rolling down a hill, and it does not require any energy. So one important thing to remember about diffusion is that it does not require ATP, or it does not require energy, because you're moving things from moving molecules from high to low. No energy is required. And then also at the same time, temperature can speed up the process. So if we increase the temperature of this glass of water, the blue dye would diffuse much faster. So we would get to that equilibrium much faster. But collectively, diffusion is the movement of molecules from an area of high concentration to an area of lower concentration until we get an equilibrium established, which basically means there's an equal number of molecules throughout the surface. Again, this does not require energy, and it does speed up at increased temperatures. So like I said, diffusion is more of a generic term that just refers to the movement of all molecules in that particular direction from high to low. Osmosis is specific to water. So osmosis is the diffusion of water. So it's moving water molecules from an area of higher concentration to areas of lower concentration, again, until that equilibrium is established. Still, since you're moving water from areas of high to low, you're again going down that concentration gradient. It does not require any type of energy or ATP. And then again, temperature increases in temperature would speed up the process. So if we look at this U-shaped test tube right here, there is like a little cell membrane here, which will allow water to pass freely through, but it will not allow larger molecules through. So over here we just have pure H2O, just pure water, and then over here we have water that does have sugar molecules suspended in it. So initially, just looking at this here, it looks like there's an equal amount of water on both sides, but that's not actually true. There's actually more water over here and less water over here. So we have to account for the space the sugar molecules are taking up. If we took all these sugar molecules out, that water level would actually go down. So based on the rule of osmosis, water is going to move from an area of higher concentration over here to areas of lower concentration in this direction, again, until an equilibrium is established. So we have that net movement of water here. By the time we get down here, I know that it looks like there's more water on this side and less water over here, but again, we have to account for the space of the sugar molecules. At this point, an equilibrium has been established, and there is actually an equal amount of water on both sides. So water will always move from an area of high concentration to areas of lower concentration, again, until that equilibrium is established, and it does not require any type of energy or ATP. So based on the rules of osmosis, there are different types of osmotic environments, and it's important for us to understand how bacteria would be affected in these different types of osmotic environments. So the three different environments are hypertonic, hypotonic, and then isotonic. So in a hypertonic environment, this would actually be the equivalent of putting bacteria in a beaker of salt water. In a hypertonic environment, there's going to be more solutes, things like sugar and salt, outside of the bacterial cell. That typically indicates that there's less water outside of the bacterial cell. So knowing that, water is going to move from an area of high concentration to an area of low concentration. So if there's more water in the cell, and less water outside of the cell, then water is obviously going to travel out of the cell, and the cell is going to shrivel, and then often the bacterial cell will die. Bacteria tend to not like hypertonic environments because it does pull the moisture out of their cells and causes them to shrivel. That's the whole reason why things like sugar and salt are used as preservatives. So if we think about like jellies and jams, obviously the sugar makes those foods taste good, but again, it also acts as a preservative. Imagine if bacteria got into a jar of jelly. Well, all that sugar creates a hypertonic environment, and it pulls the moisture out of the bacterial cells and causes them to shrivel and die. So hypertonic environments tend to be dangerous for bacteria because those sugar and salt molecules that are outside of the cell kind of pull that moisture in their direction based on the rules of osmosis. Hypotonic environments are the complete opposite. So in a hypotonic environment, this would be like putting bacteria in a beaker of just pure H2O, or pure water. So at that point, there would actually be more solutes inside of the bacterial cell and less water inside of the bacterial cell. So again, based on the rules of osmosis, water is going to move from an area of high concentration to areas of lower concentration. So based on that, water is actually going to move into the cell, and the cell is going to expand to the point where it could potentially burst. Bacterial cells are flexible with their expansion, and their cell membrane their cell wall and their peptidoglycan layer can expand to a certain point, so they can sustain a certain amount of intake of water, but if it gets to the point where they've taken in too much water, they potentially could burst. So in a hypotonic environment, water is going to be moving inward, and the cells are going to expand and potentially burst. So in an isotonic environment, we are already at a state of equilibrium. In an isotonic environment, there is an equal amount of solutes on the inside and outside of the cell, which means there's no net movement of water, and the cell remains completely unchanged. So in an isotonic environment, there's no change to the cell. There is also something called facilitated diffusion, which is still diffusion. Diffusion, again, is the movement of molecules from an area of high concentration to an area of low concentration. And that typically will happen right through the phospholipids in the cell membrane. Facilitated diffusion is still moving molecules in that direction. However, facilitated diffusion usually involves some type of protein carrier. Smaller molecules and molecules that don't have a charge can just go through regular diffusion and kind of squeeze right through the phospholipids in the cell membrane. But larger molecules and molecules that have a charge are not able to do this. So they need to be carried through some type of protein carrier. So for those types of molecules, so they're still able to move from high to low, and this does not require any type of energy. The only difference between just regular diffusion and facilitated diffusion is that facilitated diffusion incorporates the assistance of some type of protein carrier to help carry those larger molecules or charged molecules into the cell at this point.